Kitty Clinical here with our first hard medicine, hard basic science subject, biochemistry for the USMLE. And this is going to be for step 1 mainly, but you can use this to apply for steps 2 and steps 3. We're going to use some clinical correlation for some of these things, but really biochemistry, it's a lot of hard science. A lot of it is going to be on step 1. Not a whole lot of it is going to be on steps 2 or steps 3. We're going to talk about molecular, the structure of a chromosome. You probably already know about the structure of a chromosome. DNA in the condensed chromatin form fits in the nucleus. DNA loops around twice against the histone octamer, forms a nucleosome. They like to call it beads on a string. And there's different components to it. Like there's the H1 histone linker. There is that stabilizes the chromatin fibers. You know that DNA has both negative and positive charges, right? Histones are large. They have a positive charge. They usually get that positive charge from lysine and arginine. The phosphate group in DNA has a negative charge. And DNA also exists in a circular fashion in the mitochondria, right? And as you know, mitochondria comes from the mother. So when the mother and the father are conceiving, the DNA in their mitochondria actually is from the mother, because that's where the cell is coming. That's coming from the cell, right? And it's not coming from the nucleus, because the mitochondria is outside of the nucleus. Now, the interesting thing about this is that some people have, I don't know if this is a theory or if it's in practice, but there's actually an interesting case where an individual had 3 parents, right? Because they got DNA from their mother, their father, and then the mitochondria DNA came from another person, came from another female, I think. And that was a very interesting case that I either read up on, or it was like a real case. And I will definitely pull it up and share it on this YouTube channel once I find it. I am interested in feeling out some interest in very interesting cases in medicine, or freak accidents, things that are just really weird, right? And so that's one of the interesting things about biochemistry and molecular, is that you get these weird things that are just very interesting. Now, as you know, there's different parts of a cell cycle, the phases, right? G1, S, G2, and M. G1 being growth, S being DNA synthesis, also synthesizes histones during the S phase, G2 being growth and preparation for mitosis, and M being mitosis, cell division, right? So that's going to be the basic concepts for our structure of the DNA, and just about the special jargon of chromatin and the different things that you need to know. We're going to jump into that right now. Basic concepts. You know that there's methylation and there's acetylation, right? So methylation, think of it as deactivating or muting the signal on the DNA. So if you're methylating something, you're inactivating it. You're stopping it from happening. You're stopping that part of the DNA from generating the proteins and what have you to do whatever it's got to do, right? Acetylation is activating. You're activating the signal of that DNA sequence, right? So that's going to be some basic concepts. You know, heterochromatin is the condensed form, and because it's so condensed, it's so much darker. It's hard for transcription to happen, which means obviously there's more methylation, there's less acetylation, and it's just structurally it's not accessible. It's what some people call it sterically inaccessible, right? So that's going to be the heterochromatin. You may also, like in another example would be like Barr bodies, BARR, which are inactivated X chromosomes visible on mainly the periphery of the nucleus. You've got euchromatin, which is the opposite of heterochromatin, less dense. It's lighter on imaging, right? It's transcriptionally active. You can access it sterically, which means from a structural standpoint, it's easy to function. It's easy for you to get into it. It's euchromatin, right? It's good chromatin. And then there's, we talked about DNA methylation, which you can see in aging, imprinting, cancer. You can see it in that transposable element repression, lionization, which is X chromosome inactivation. So DNA is methylated when it comes to imprinting. You've got something called CPG islands, which is where methylation is within the gene promoter, typically silencing the gene transcription. CPG, it's capital C, lowercase p, capital G, methylation. It basically stops the DNA from being active, right? That part of the DNA is basically not usable, and you can kind of see the dysregulation in DNA methylation in other pathologies such as Fragile X syndrome, right? Histone methylation is another thing that we talked about before. Methylation being, suppressing, and when it comes to histones, it's actually a reversible transcriptional suppression. So it can actually cause activation depending on the location of the methyl group. So again, we talked about lysine and arginine being the residues of histone that can be methylated. Acetylation being activated, right? So you're removing the histone's positive charge. The DNA coiling is lessened and it's being relaxed. You can increase the transcription. You can actually see histone acetylation in thyroid hormone receptors, altering the thyroid hormone synthesis. That's actually an acetylation process. You can see histone acetylation making DNA. Basically, it's making it more active, right? Acetylation, we've kind of hammered this down here. And then histone deacetylation, removing the active group, which means you're decreasing transcription. You're tightening the DNA coil, right? Because that's what remember, that's the function of the histone, right? Is involved in that property of the DNA. So that can also the histone deacetylation, you might see that in Huntington's disease, which as you know is a very high yield topic. For some reason they love Huntington's. They love all of those diseases that you very rarely see in the clinic or in the outpatient or inpatient setting even. So anyway, that's going to be it for this first segment of biochemistry. Hope you enjoyed it. I will see you in the next one.